Well, hello, 2421 students. I want to pick up again and complete the rest of chapter 18. So this is going to be part two. And if you recall from last time, what I was talking about was the genetic basis of graft rejection. And remember, graft rejection is basically a function of cytotoxic T cells. They're always checking these MHC proteins, MHC class 1, also called human leukocyte antigen. And if they see strange MHC proteins, well, they go nuts and they'll obviously try and kill it. Now, when we look at the genes that are involved in this, here's here's the A and the B and the DR genes. There's actually 6 alleles. There's 6 of them. There's 6 alleles, but that's a total of 12 genes, right? Because you get 6 from this parent and 6 from this parent, and this is confusing because they're only showing 3, but they're actually 6. There'd be like HLLCW, HLLDQ, and DP, and so on. They're just not showing that because when they do tissue typing, they're really looking at these 3. These are the 3 that are sort of out there and the sort of drives the cytotoxic T cells nuts if they're not the same as as your own HLA proteins. So this is what they key on when they actually do tissue typing. Although there are actually 6. So there should be 3 more there and then 3 more there. And I just want to mention that. And practically speaking, it would be very difficult to get all 12 alleles identical. That's just not gonna not all 6 alleles identical or all 12 genes identical, right? So, this is 2 genes, right? And then another 2, 2, 4, 6, and then there'd be another 3 down here and another 3 there to give you a total of 12. Okay. So, what's going on is, of course, if you're a child of your parents, you're going to inherit, these from one parent and these from another, right? Now, the thing is, these are highly, highly polymorphic. And what that means is there are many, many different alleles for each of these genes. So you can see you have 9 in one, and you can actually have hundreds of them. They're the most polymorphic genes known, as a matter of fact, in terms of alleles. And so consequently, if you have 2 parents, and you should, then you're clearly going to have a different alleles here at these various gene locations. I mean, that's just inevitable. Now, what they're trying to do is they're trying to match 3 alleles. In other words, they want three of these alleles, A, B, and DR, to be the same. So, where do you find that? Not in the general population, because the chances of the general population actually having 3 identical alleles to you would be very, very unusual. In other words, you'd have to have a 9 and a 1, and then you'd have to have a 4 and a 7, and then you'd have to have a 2 and 11, and that's just not very, very likely. But for your siblings, it's not that unlikely, because your siblings are going to get these same things, right? And so you have about 1 in 4 chances of having a sibling who's going to have this same haplotype here that you have. Haplotype meaning that, well, you get half from your mother and half from your father. So these are the haplotypes there, right? I know that's a lot of red there that I'm drawing, but that's the case. So you need that kind of match. You need those 3 alleles to be identical with the person whose organ you want to use. So could your mother or father donate an organ to you? And the answer to that is no. That'd be terrible donors because, well, say your mother might have, these particular proteins on their cells, but they certainly wouldn't have the father's protein. And remember, you need 3 of these to match. You would need 9 and 1 and 4 and 7 and 2 and 11 and so on. And of course, neither your mom or dad are going to be a match. They're gonna have half that you really need. So siblings are really the key here. You have about 1 chance in 4 of having a sibling. If you have 4 siblings, probably one of them is going to have that match, and that's really great if you're on good terms with your sibling. Oh, I need a kidney, so please fork over. But that's about it. And then finding a match in the general population is very unlikely. You have to go through a lot of them, and that's what they do. That's what they do when they try and do tissue typing and so on. Which is why it takes so long to actually find a match. Because you have to find a match, and the person must have died accidentally, and their organs must have been harvested, and it must be ready to go. Right? Unless you're in China. Because in China, if you're a prisoner, the first thing they do is do tissue typing, and I don't even want to go there. It's pretty grim. But let's move on here. So a 6 antigen match, right? Those 3 alleles, which are 6 genes. That's what you want. That's what you're looking for. That's what they mean when they say tissue typing. Now, there are other things involved in tissue typing. If you watch that video, you'll see. But basically, that's the most important thing, because cytotoxic T cells are essentially what's destroying your organ that you get, and that's pretty important. So I hope that's clear. And watch the video. I think it will be clear to you if you do that. But if you ever need an organ, it's something to to think about. And if you know someone who needs an organ, you can talk to them about the HLA bit and sort of inform them on that. So what about graphs? Let me look at my notes here. What about graphs? Well, there are certainly autographs, and I'm obscuring this picture, but this is a guy with an autograph. He's getting a hair implant there. You see these little plugs that many guys get to try and look young or something when they're in their 40s and 50s. These graphs are superb because, of course, you have the HLA, all the LLs are identical. So this is some of the first kidney transplants were done, for example, with identical twins, and they were done successfully because you don't have to worry about rejection. This is much more difficult because you have to at least try to find a match, and this is really impossible, these xenotransplants from like a pig to a human or whatever. And that's because not only are all of the MHC class 1 proteins different, all of them are different, but they also have other proteins on their surface that really drive your immune system crazy. They have all kinds of funny proteins that we just don't have, and this leads to all kinds of problems, as a matter of fact. So you get this hyperacute rejection within minutes, as a matter of fact. 30, 40 minutes, you're rejecting your organ. But you can do grafts pretty easily, and some of the most astounding grafts have been done a long time ago. If you've watched this video here, this is a head graft, a head being transplanted from one body to another. And you might think that's difficult to do, but it's really not. That was done in the early 60s. Of course, they did it with monkeys, not humans. But nonetheless, it is technically pretty easy to do the grafts What's hard to do is to do, the grafts that aren't rejected. And even with drugs, that's a problem. Now, there's something called graft versus host. This is transplanted bone marrow. This is where, when we do graft versus host, I'm looking at my notes here in my head. Brother. This is usually done with leukemia, where you destroy your bone marrow and then put in someone else's bone marrow in there. This is the notion, but you'll always have leftover T cells and the lymph nodes and so on. And the thing is, but the thing is, these T cells coming out of the bone marrow may start attacking you. It's graft versus host, and this is a real problem. So, but this is really kind of graft versus host. It's not a big deal because it's really only leukemia and so on. I really like this this figure. It depicts the the various kinds of grafts quite well. And so take a peek at that. It turns out that if we consider grafts and so on, there are certain privileged sites in the human body, and these are sites where you don't really see much rejection. I can take the brain and transplant. Say you had a stroke, you have some dead brain tissue, cut it out, and you put new brain tissue in there. That's perfectly possible. And, you don't really get much rejection, simply because you don't have a lot of cytotoxic T cells in your brain. Brain cannot tolerate much inflammation. It can't tolerate much, cell death here. It just can't. So that's not a big problem. However, if you put in brain tissue, it's not going to reconnect, or even if it does, it'll just sort of reconnect willy-nilly, and it won't be functional, so there's really no point in doing that. Cornea, the cytotoxic T cells can't get to it, so you can certainly have corpse corneas implanted there, put onto your eye. It's not going to hurt anything. It's interesting that the testes here, the testes are a privileged site. In other words, your immune system doesn't bother your testes very much. And the reason is and the eyes as well, and the eyes are kind of obvious. If you get a lot of inflammation and cytotoxic T cells start killing, retinal cells willy-nilly, then what's going to happen, of course, is you'll go blind and you'll drop out of the gene pool. People with those kinds of immune systems don't last in terms of evolution. But the same thing is true with testes. Because testes, if you get a lot of inflammation there, if cytotoxic T cells start killing those cells, what happens is you drop out of the gene pool pretty quickly. You disappear. So you don't want immune systems that screw up your testes, so to speak. And in fact, some of the first transplants that were done with testes. And not only that, they were xenografts. That is to say, people, and this is kind of fascinating. If you click on that, look at the hyperlink. This is about John, I'll just abbreviate his name here, John R. Brinkley. Now, John R. Brinkley is a doctor who became famous by putting goat testicles in scrotums. I mean, he did. And it seemed to work. It was sort of the early version of Viagra, and the goat testicles apparently secreted enough androgens or testosterone to actually, perk up guys. It seemed to have a real effect upon them. But the thing is, you didn't have to worry very much about rejection of the goat testes because it was the testicles and their privileged side. So anyway, I find John R. Brinkley kind of fascinating, so you should watch that. And I might ask you a couple of questions about John R. Brinkley. He was an interesting character back in the day here. It's quite a while ago, to say the least. But anyway, there are a few other sites like that, and it's an interesting notion, this idea of privileged sites. Immunosuppression, there are a number of drugs that do that. The first one that I do want to talk about is cyclosporin. This was the first effective immunosuppressive drug, and it's suppressing IL-2, which is needed for cytotoxic T cell activation. So you see them here. And that's what you're trying to suppress more than anything else, although there are drugs that suppress both B and T cells. But cyclosporin was derived from a fungi, and this picture here, that's the fungi, as a matter of fact. So fungi are really kind of interesting in that they do all kinds of interesting biochemistry, including compounds that suppress our immune system. Let me go to the next one here. The book has a little bit on AIDS and immunosuppression in general. Now, there are some genetic diseases here that causes the suppression of the immune system. I talked a little bit about chronic granulomatous disease when I was talking about phagocytosis, and this is the fact that they don't produce too much NADPH, and I'm sure you remember what that does, right? It doesn't produce too much of that. People have genetic problems who don't make much of that. And the thing is, what does NADPH do? It takes oxygen and converts it into an oxygen-free radical. And you need that to make things like H2O2 and bleach and so on. And if you don't have that, you get all kinds of terrible infections. Now, you can see that there are other kinds of symptoms like DeGeorge's syndrome here. This is where your thymus, it says lack of T cells and no cell-mediated immunity. This is where your thymus doesn't develop correctly because of genetic problems. So, you have a terrible problem here. Usually you get thymus transplants and so on. The most terrible one is where you have both a lack of T and B cells and you just die essentially pretty quickly. Also, the book talks a little bit about HIV. I did go over the HIV life cycle, if you recall, when I talked about viruses, so I do expect you to know that. I'm not going to go through it here. There's no point in doing that. But again, you see that HIV, and what does HIV do? Well, it screws up your immune system because it infects T cells, T helper cells, and T helper cells. If you're, it's keying on the receptor is CD4, of course, and we know that all T helper cells have CD4 co-receptor for what? For MHC class 2, right? That's what that CD4 is there for, because the T cell receptor binds to MHC class 2. But why does it do that? Because it has this co-receptor called CD4. And that's actually what the virus is attacking. Now, it does need co-receptors, these cytokine receptors normally, like CCR5 and that kind of thing. And you do see it here. I think I can barely make this out here. CD4. I guess it's really not showing that. Not really. Here's CXCR4 as one of the co-receptors there that it needs to bind to. There are a couple co-receptors that it can use. Oh man, is that tiny? CXCR4 is one of them, and CCR5 is another. And it just allows it to fuse, gets into the cell, integrates into the genome. It screws that. Well, first of all, it may or may not kill the T helper cell, but even if it doesn't kill it, it screws up the signaling mechanism of the T helper cell, and that really screws up your immune system. So, just a couple of factoids that I thought I would mention about that. These numbers here can vary quite a bit depending upon where you are in the infectious stage of the of AIDS or HIV infection. In other words, at the end, your immune system kind of collapses, and these numbers would increase enormously. But there's quite a while where these numbers are as low as that, or even lower, and way lower in terms of blood. But it does vary quite a bit depending upon where you are in the virus here. If you're like this guy has Kaposi sarcoma, he's in bad shape, and his viral load in his blood and semen, saliva would be enormous. If you don't have symptoms, it's much less. Let me see here. One little factoid I do want to mention: if you have less than 200 T helper cells per microliter, which is not much, then you certainly would start getting these diseases. And so on as you fall underneath that. If you have that, you would be officially diagnosed as having AIDS. And then this is a little bit of where the virus is, or how did it come about? And I guess both of those are actually pretty watchable. Of course, it came about in Africa. And it just did. So, take a peek at that. I think both of these are interesting. I might ask you a question or two on that. And that's the end of that. So it's only 23 minutes, but last night when I was doing this, I really pooped out. So, take a peek at that, and we'll continue with our next chapter soon, because I have to finish all this stuff up. But I'll, call it a night, because I'm at night now, and I'll wave goodbye, and see you next time.